Hello, my dear students. Welcome to a new episode of biology. Today, we will talk about traits and probability. The key concept for today's lesson is the inheritance of traits follows the rules of probability. The main ideas: Punnett squares illustrate genetic crosses. Number two, a monohybrid cross involves one trait, and the dihybrid cross involves two traits. Then, heredity patterns can be calculated with probability. So let's start. Shortly after Mendel's experiments became widely known among scientists, a poultry geneticist named R.C. Punnett developed the Punnett square. This is the Punnett square. The Punnett square is a great system for predicting the possible genotypes of offspring, as you can see. So the parent alleles are written on the axis, and the gray boxes, this is for the possible genotypes of the offspring. The Punnett square is a great system for predicting all the possible genotypes resulting from a cross. Genotypes, this is the genetic makeup of the organism. The axis of the grid represent the possible gamete genotypes for each parent of each parent. The grid boxes show all of the possible genotypes of the offspring from those two parents. Because segregation and fertilization are random events, each combination of alleles is as likely to be produced as any other. By counting the number of squares with each genetic combination, we can find the ratio of genotypes in that generation. If we also know the how the genotype corresponds to the phenotype, or how it contributes to the phenotype of the organism, we can find the ratio of phenotypes in that generation as well. So from the Punnett squares, we can figure out or find out the ratio of the genotypes and also the phenotypes. Each gamete gets one of the alleles, yes, for sure. Since each parent contributes only one allele to the offspring, only one allele from each parent is written inside each grid box. Fertilization restores the diploid number in the resulting offspring. This is why each grid box has two alleles, one from the mother and one from the father, to restore the diploid number. Since any egg has the same chance of being fertilized by any sperm cell, each possible genetic combination is equally likely to occur. So what's a monohybrid cross? A monohybrid cross is the cross that examines the inheritance of one specific trait, only one specific trait. We have three example crosses ahead to illustrate how Punnett squares work and to also highlight the resulting ratios for both genotype and also the phenotype. Keep in your mind that genotype is the genetic makeup of the organism, while phenotype is the physical appearance of that organism. So the first one is homozygous. Suppose you cross a pea plant that is homozygous dominant for purple flowers with a pea plant that is homozygous recessive for white flowers. To determine the genotypic and phenotypic ratios of the offspring, first write each parent's genotype on the axis. F capital for the purple flowered plant and F small for the white flowered plant. Then every gamete from the purple flower plant contains the dominant allele F capital, and every gamete from the white flowered plant contains the recessive allele F small. So 100% of the offspring have the heterozygous genotype F capital F small, and 100% of the offspring have purple flowers because they all have a copy of the dominant allele. As you can see in this square, the parents, the homozygous dominant parent, F capital, we said that every allele is written above. And to the next side, this is the homozygous recessive parent, F small. So in each box, you distribute only one allele from each parent. So this F1, this F capital will be here, distributed here and here. And the second F capital allele will be here and in this box as well. Then when you distribute the F small alleles, F small, and this one F small, so all the offspring, they are F capital, F small, so they are all purple, and they are all heterozygous. Okay, then the second example is heterozygous. As you can see here, both the parents are heterozygous for the trait. So they have purple flowers, but they are heterozygous. One allele dominant and one allele recessive. This is F capital F small. And the other parent F capital F small. So when you distribute it, the first square will be F capital F capital. This is homozygous dominant. And the second one, F capital F small. The third one, F capital F small. They are both heterozygous for the trait, and the last one is F small, which will be homozygous recessive. Okay, in the previous Punnett square, you can see a cross between two purple flowered pea plants that are both heterozygous, F capital F small, one allele dominant and one allele recessive. From each parent, have the offspring receive a dominant allele, F capital, and the other half receive a recessive allele F small. Therefore, 1/4 of the offspring have a homozygous dominant genotype, which is F capital, as we saw, this is it, and half have a heterozygous genotype, which is F capital, F small. Here they are, this box and this box. And 1/4 have a homozygous recessive genotype, F small. This was the last square. This is the white flower. Okay. Both the F capital and F capital F small genotypes result in purple flowers. So this is the same phenotype. Only the F small genotype results in white flowers. So the genotypic ratio is 1 to 2 to 1 of homozygous dominant, 1 to heterozygous, which are 2, to homozygous recessive, which is 1. But the phenotypic ratio is 3 to 1, which is 3 purple to 1 white flowers. Then the third example is heterozygous homozygous. This is different. So heterozygous parent, F capital F small, and the homozygous recessive parent, F small. In this case, what would happen? We will distribute the alleles on the axis, then distribute the gametes, one allele from each parent in the squares, as usual. So the first one will be F capital F small, the second square, F capital F small, the third one, F small, and the fourth, F small. So as you can see, it is 50-50 or 1 to 1. Okay, finally, suppose you cross a pea plant that is heterozygous for purple flowers, F capital F small, with a pea plant that is homozygous recessive for white flowers, F small. As before, each parent's genotype is placed on an axis. From the homozygous parent with white flowers, the offspring each receive a recessive allele, which is F small, yes. From the heterozygous parent, the half of the offspring receive a dominant allele, F capital, and the other half receive the recessive allele, F small. So half the offspring have a heterozygous genotype, F capital, F small, while the other half have a homozygous recessive genotype, F small. So half the offspring have purple flowers and the other half have white flowers. The resulting genotypic ratio is 1 to 1, heterozygous to homozygous recessive, but the genotypic ratio is also 1 to 1, purple to white. Then we come to another process named test cross. Test cross, from its name, it's a cross that is used in a test. So what is this? Suppose we didn't know the genotype of the purple flower in the cross above, which was heterozygous homozygous. This cross would allow us to determine that the purple flower is heterozygous, not homozygous dominant. So what is a test cross again? A test cross is a cross between an organism with an unknown genotype with an organism with the recessive phenotype. Why recessive phenotypes? Because the recessive phenotype is always homozygous recessive. So we already know what is the genotype of this one. Okay, so the organism with the recessive phenotype must be homozygous recessive for the trait to appear. The offspring will show whether the organism with the unknown genotype is heterozygous or homozygous dominant. Then we come to the second type of cross, which is a dihybrid cross. It involves two traits. All of the crosses discussed so far have involved only a single trait. However, Mendel also conducted dihybrid crosses. What is a dihybrid cross? It's a cross that determined the inheritance of two different traits. Mendel wondered if both traits would always appear together or if they would be expressed independently of each other. So Mendel performed many dihybrid crosses and tested a variety of different combinations. For example, this is the one that we will discuss. Mendel would cross a plant with yellow round peas with a plant with green wrinkled peas. So this is the seed color and the seed shape. These are two different traits. Remember that Mendel began his process with purebred plants. So the first generation offspring, F1, would all be heterozygous and would all look the same. In this example, the plant would all have yellow round peas. When Mendel allowed the F1 plant to self-pollinate the plants of the first filial, which were all yellow round peas, to self-pollinate, he obtained the following results. 9 plants have yellow round seeds, 3 have yellow wrinkled seeds, and 3 have green round seeds, while 1 have green wrinkled seeds. So what is this? Mendel continued to find this approximately 9:3:1 phenotypic ratio in the F2 generation, regardless the combination of traits. From these results, he realized that the presence of one trait did not affect the presence of another trait. And then he concluded the law of independent assortment. So what is the law of independent assortment? It states that allele pairs separate independently of each other during gamete formation or meiosis. So the alleles for each trait, they separate independently of each other during gamete formations or during meiosis. That is, different traits appear to be inherited separately, one by one, separated from each other. Drawing a Punnett square for a dihybrid cross is the same as the Punnett square for a monohybrid cross, but it will be bigger, except that the grid is bigger because 2 genes or 4 alleles are involved. So we have 4 chances. For example, suppose that you cross 2 plants with yellow round peas that are heterozygous for both traits. Now we have 2 letters because we have 2 traits, and each one for each trait with the 2 alleles. So if Y for the yellow seeds, this is Y capital. So the green seeds will have Y small allele, and then R capital for round seeds and R small for wrinkled seeds. So now the 2 plants that are heterozygous for both traits, they have Y capital Y small, R capital R small. Okay. The 4 allele combinations possible in each gamete, Y capital R capital, Y capital R small, Y small R capital, Y small R small, are all used to label each axis. Each grid box can be filled in using the same method as that used in the monohybrid cross, the same. A total of 9 different genotypes may result from the cross in this example. However, these 9 genotypes produce only 4 different phenotypes. These phenotypes are yellow round or yellow wrinkled, green round or green wrinkled, and they occur in the ratio 9:3:1. Also, note that the 9:3:1 phenotypic ratio results from a cross between organisms that are heterozygous for both the traits. They have the 2 alleles, the dominant and recessive, for both the traits. The phenotypic ratio of the offspring will differ from 9:3 to 3:1 if one of both the parent of organisms are homozygous for one or both the traits. So this phenotypic ratio is only for the heterozygous parents for both traits. And here is the grid box. As you can see, here we have on each axis, this is the apparent that is homozygous, that is heterozygous for both traits, and in this axis also. So what are the possible combinations? Y capital R capital and Y capital R small, then Y small R capital and Y small R small on both the axes. Then when you combine these in the boxes, as you can see, how many of the offspring they have homozygous dominant, or they have the yellow round seeds? 1, 2, 3, 4, 5, 6, 7, 8, and 9. They have one allele dominant for each trait. This is the yellow round. And how many for the yellow wrinkled? 1, 2, 3. So this is the second 3. Then how many have green round seeds? This 1, 2, 3. And how many have green wrinkled seeds? Both the traits recessive 1. So this is the ratio 9, 3, 3, 1. Okay. Then heredity patterns can be calculated with probability. What is probability? It's the likelihood that a particular event will happen. Probability predicts the average number of occurrences, not the exact number of occurrences. So how can we calculate probability? It equals the number of ways a specific event can occur over or divided by the number of total possible outcomes. What is this? Let's see. Suppose you flip a coin. The number of total possible outcomes is 2, whether the heads up or the tails up. There is no other chance. The probability that it would land heads up is half, or 1 out of 2, and the probability that it would land tails up is also half. Next, suppose you flip 2 coins together at the same time. How one coin lands does not affect how the other coin lands. Yes, for sure. To calculate the probability that 2 independent events will happen together, multiply the probability of each individual event. What is this? For example, the probability that both the coins will land heads up is half times half equal 1/4 or 1/4. Here it is. So this is the same as the Punnett square. This is heads up for one coin, and this is tails up for the same coin, and this is for the second coin. Then each probability have 1/4 the chances to happen. 2 heads up, 1 head, 1 tail, 1 head, 1 tail, and the tails up. These probabilities can be applied to meiosis. Suppose a germ cell undergoes meiosis in a plant that is heterozygous for purple flowers. So this is like it's F capital F small. For example, the number of total possible outcomes is 2. Yes, because a gamete could get a dominant or a recessive allele. The probability that the gamete will get a dominant allele is half the chances, or 1 out of 2. And also the probability that it will get a recessive allele is also 1/2. If 2 plants that are heterozygous for purple flower fertilize each other, the probability that both egg and sperm have a dominant allele is half times half equals 1/4 or 1/4. So also the probability that both have a recessive allele is also 1/4. There is also a 1/4 chance that a sperm cell with a dominant allele will fertilize an egg with a recessive allele, and also vice versa. Or that a sperm cell, the sperm is with the recessive allele, will fertilize an egg with the dominant allele. So these last 2 combinations are basically the same. In either case, the resulting plant will be heterozygous. So the probability that a pea plant will be heterozygous for this trait is the sum of the probabilities. 1/4 plus 1/4 equals half. I hope you understood this part. See you in the next episode. Thank you guys, and goodbye.